DNA structure and replication. What is DNA? DNA is a carrier of genetic information that codes for all our physical traits or phenotypes. Inherited alleles code for things like eye color, hair type, and height. DNA is a complex macromolecule made up of 3 parts. The base component is a deoxyribose sugar. The second part is a phosphate group, which is attached to the deoxyribose sugar. The third part is a nucleotide base. There are 4 nucleotide bases in DNA: adenine, guanine, thymine, and cytosine.
Adenine and guanine are purines, while cytosine and thymine are pyrimidines. Adenine always pairs with thymine, and cytosine always pairs with guanine. This is called Chargaff's rule. Guanine and cytosine always have triple hydrogen bonds, and adenine and thymine always have double hydrogen bonds. Overall, the DNA strands run opposite to each other. There's a 5' and 3' end for each strand, and they run anti-parallel to each other. This results in a double helix shape.
There are some differences between prokaryotic and eukaryotic DNA. Eukaryotes, such as humans, have a linear DNA. It is not one continuous loop DNA like prokaryotes. In addition, DNA in prokaryotes has only one origin of replication, while in eukaryotes there are multiple. Furthermore, in prokaryotes, DNA is free-floating in the cytoplasm, while in eukaryotes it is organized in the nucleus.
Whenever a cell divides, DNA has to replicate. Replication is a very complex process. DNA replication is semi-conservative, meaning each strand of DNA acts as a template strand for another DNA. The first step is the creation of a replication fork. DNA helicase, an enzyme, destroys the hydrogen bond between the bases and unzips the DNA. DNA topoisomerase, another enzyme, relieves tension ahead of the replication fork by untangling the helix. Now, DNA is ready to replicate.
DNA replicates in 2 different fashions, the first of which is a leading strand, and the second of which is a lagging strand. Now, we'll focus on the leading strand. In the leading strand, DNA replicates in the 5' to 3' direction. In step 1, RNA primase puts down an RNA primer on the 5' end of the DNA. DNA polymerase III adds on DNA nucleotides to the RNA primer and elongates the new leading strand. After some time, DNA polymerase I replaces the RNA primer with DNA molecules. Finally, DNA ligase joins the molecules together.
Now the lagging strand. The lagging strand runs opposite from the leading strand, so DNA replication will not be continuous. RNA primase puts down an RNA primer on the 5' end. DNA polymerase III elongates the lagging strand starting from the RNA primer. Then another RNA primase goes behind the original fragment and puts down another RNA primer. DNA polymerase III elongates this fragment, and the cycle repeats many times.
Eventually, DNA polymerase I replaces the RNA with DNA molecules. Then DNA ligase smooths out the Okazaki fragments by creating bonds between the former primers and the new DNA. Each of these fragments are called Okazaki fragments after Reiji Okazaki, a Japanese scientist. Overall, this process of replication results in 2 identical strands of DNA.